Hello everyone, and a warm welcome back to our Evolution and Ecology seminar series. My name is Elizabeth Duxbury, and I'm a postdoc in the research group of Alexei Maklikov at the University of East Anglia, and I'll be hosting today's session. Myself and our wonderful team of co-organisers, Andreas, Ulia and Wouter, are very grateful for the enthusiasm from the research community for these seminars, and it's been a pleasure to host such a variety of great minds so far. So thank you all for your fantastic engagement in the series. Before I introduce today's speaker, I'd like to remind you all that there will be a Q&A session directly after the talk. So please post your questions in the dedicated Slack channel and upvote questions that you'd like to hear. We aim to keep this whole session to 1 hour in length. So it is my great pleasure to introduce today's speaker, Professor Anna Qvarnström, from the Department of Ecology and Genetics at Uppsala University in Sweden. Anna's research explores how new species are formed and maintained, and she is particularly fascinated by the ecological and behavioural drivers of speciation, as well as the barriers to hybridization. Anna has conducted extensive research using two enigmatic field study systems. Firstly, using long-term studies with the collared and pied flycatchers in the Åland Islands of the Baltic Sea, Anna is investigating the impact of climate adaptation on speciation in the hybrid zones. And secondly, Anna's research explores the role of anti-predator coloration in the strawberry poison frogs of a Caribbean archipelago off the coast of Panama. Anna's research combines an innovative combination of long-term field work, built-in pedigrees, and genome-wide association studies. More recently, she is also addressing fascinating questions such as the genetic and neurological basis of song learning and its role in speciation, in addition to the impact of avian malaria on interspecies interactions. Anna has received several awards throughout her career, as well as holding a prestigious European Young Investigator Research Fellowship. Today, Anna will present her talk entitled Climate Adaptation and Speciation. So thank you for accepting our invitation, and over to you.
Yeah, thank you for that very nice invitation and presentation of my work. So today I will be talking about climate adaptation and speciation, and I will mostly focus in on work that my group has done on the Flycatcher system. So I'm truly fascinated at looking at biodiversity out in the world, and I want to understand how it can origin. And the only process that can generate new biodiversity is speciation. And speciation can in most cases be seen as a split of one original species into two new species. And when this happens, these new species can develop independently and follow their own evolutionary pathways. And when we focus in on sexually reproducing animals, this often occurs through build-up of reproductive isolation, because if they don't become reproductively isolated, they can't really follow independent evolutionary pathways. So when we work on speciation in animals, we want to figure out how they can become reproductively isolated from each other. So we can look at different types of isolation. We can focus on isolation that happens before the zygote is formed, so prezygotic isolation, how animal species become reproductively isolated by processes that happen before they actually mate or before the zygote is formed. Then we can also look at postzygotic isolation, which happens after the zygote is formed. So we could have hybridization happening, but the hybrid could do poorly in the ecological environment of the two parental species and therefore be selected against. Or the hybrid could have problems in finding mates. Or if the two different divergent populations are so genetically different, they might not even be able to function well together, so the genomes become incompatible. And then the hybrids might be sterile or not even viable. But that would be at a very late stage of the speciation process normally. So when we study speciation, we can be interested in different types of questions. So one type of question would be how? What are the mechanisms that actually pushes two populations apart and make them reproductively isolated? Is it through natural selection or sexual selection or something else, like mutations? That could also happen just through drift. That would be the mechanisms. But we could also be interested in questions related to where speciation can happen. So if you look at these examples of fishes, could they speciate when they are within the same river? Or do they need to be geographically isolated, at least in the beginning of the process? We could also be interested in questions related to when speciation happen, which is, how long it will take. That is actually the length of the different branches in the tree of the species. If we want to link climate and climate change to speciation processes, most often this has been linked to the question where. So we have the classical work by Hewitt showing that hybrid zones often co-occur in a particular pattern that is compatible with the thought that populations have been pushed back during the Ice Age. So this is a picture of Europe. And when they then have recolonized Europe, they do so from different directions. And when they spend time in isolation, in geographic isolation long enough, they might not be able to mate and produce viable offspring in the end. So then you have a speciation occurring in allopatry. And climate is important because it sets the stage for where species can occur. When we're interested in the questions how, that would be related to how natural and sexual selection pushes populations apart. Most of the work has been done in relation to morphological changes associated with traits that influence what type of resources these animals uses when they eat. So one classical example of this type of ecological speciation would be the Galapagos finches and the classical work by Rosemary and Peter Grant. And they have focused in on different sizes of the beaks of these birds that relate to the size and type of seeds that they can eat. And then when two different species of these birds co-occur, there would also be like competition over the similar seed sites, which also pushing them further apart. So we have character displacement, which further sort of reinforce the speciation process. And we have two distinct phenotypes associated in using different niches. A classical example of ecological speciation. When it comes to climate adaptation in this context, it's a little bit more complicated to look at climate adaptation because it often deals with a physical tolerance to different climate factors such as temperature and moisture. And physiology can be more tricky to measure than our morphological traits, especially if we look at, for example, museum samples. It's impossible to do that. So there's been less attention paid to climate adaptation as a driver of speciation in this context in relation to the questions how. Which brings us to the outline of my talk today. So there will be two different parts of the talk, and I will zoom in on the flycatchers as I mentioned before. The focus would be ecological speciation with focus on divergent climate adaptation in these two bird species and how that could lead to different sources of reproductive isolation. And the two different sources I will zoom in on is first divergence in phenology, how that could lead to temporal isolation, which is an example of prezygotic barrier to gene flow. And then in the second part, I would talk about divergence in physiology and how that could lead to genetic incompatibility, which would be an example of a polyzygotic barrier to gene flow. So why do we stay with birds? So birds are classical examples used in speciation research because we can look at very many different components of the speciation process. Birds has a very wide distribution across the globe, so we can look at various sources of ecological speciation. They have a very high diversity, and they are very charismatic. So there are very many reasons to study birds if you're interested in speciation. The study system I use is pied and collared flycatchers, which are good models to study reproductive barriers because we can follow them individually. So we have pied flycatchers where you can see the distribution shown in red here on this picture here. And then we have collared flycatcher, which are more sort of southern distribution in Europe. And then we have areas where the two species co-occur and hybridize. An older hybrid zone in Europe and a young one on the Swedish islands of Gotland and Öland. And I study these species on Öland. You can see a zoom in on Öland here, and the different red dots are different nest box areas. And both these species prefer to breed in deciduous forests. So we put up a lot of nest boxes, and that attracts the birds because they like to breed in those. The males arrive a little bit before the female in the spring. So both these species migrate to Africa during the winter. And then they come to Sweden in the spring in May. And the males arrive before the females and start to take up suitable breeding territories. And then the females arrive approximately a week later, and it's a female who builds the nest and incubate the eggs, but then both males and females help in feeding the offspring together. And they only spend 2 weeks approximately in the nest, and then they're ready to fledge. So from hatching until fledge is only 2 weeks, which is amazingly fast. On Öland, we have approximately 79% collared flycatchers and 60% pied flycatchers. And this is because collared flycatchers are dominant, so they tend to push away pied flycatchers from the most preferred breeding sites. And approximately 5% is mixed pairs. So hybridization is quite common. Climate change in relation to phenology in birds have attracted a lot of attention. So I just list some of the papers here. And many of these papers have focused in on how a changed climate can lead to changes in phenology. And the birds need to time their phenology according to their food. And if they eat insects and insect larvae, they need to time their laying date so that the offspring hatch at a date when there are plenty of food around for them, so the parents can bring food to the nestlings. And the caterpillar themselves, they need to time their own breeding to the phenology of the trees. And the trees are influenced by the climate. When we talk about phenological mismatch, what happens then is, as I mentioned, the birds need to match their breeding so that their offspring sort of are mostly hungry when there are most caterpillar larvae food around, which is very like nutritious food for the birds. But if there is climate change and the caterpillars start to emerge earlier, then the birds also need to breed earlier. And even if the birds breed earlier than they used to do before, they could still become mismatched with a caterpillar peak if they don't breed early enough. So that would be a big problem for them, of course, because then their reproduction would fail. So how do the flycatchers react to climate change? This part of the work has been mainly done by a postdoc of mine, Päivi Sirkiä, but also Aaron McFarlane, who did a PhD with me earlier. And this is Päivi in the picture. So to just zoom in on the results, so because we monitor the birds in great detail, We know when they start breeding, when they lay the eggs, and when their offspring hatch, and when they fledge. And as you can see in this result, when there is a warmer spring, collared flycatchers breed much earlier, so their laying date is advanced when it's warmer. It's the same happening in pied flycatchers, but to a lesser extent. So when we look over time, so we've been studying these birds almost in 20 years at the Öland. So what happened over this period is that they breed earlier and earlier. Both species do that. But collared flycatchers respond much stronger. So there would be a divergence in breeding time over the years. So why do collared flycatchers respond stronger to climate change as compared to pied flycatchers? They are very similar, so they eat the same type of food. Both of them prefer juicy caterpillar larvae, but they are quite generalistic. And they have quite similar feeding techniques as well. But there are some slight life-history differences between the two species. So collared flycatchers are slightly more aggressive, which makes them dominant when they fight over territories. And therefore they push away pied flycatchers from the most preferred breeding sites in the deciduous forest. They also have nestlings that beg a little bit more aggressively, so they're more hungry, and they lay smaller clutches. But even if they lay smaller clutches, their nestlings are still more sensitive. So in cases when they sort of miss this peak in caterpillar abundance, they are much more sensitive to that than pied flycatchers. So the nestlings are more likely to die. And the same is true when they breed in a less sort of favorable environment. If they breed in more mixed forests, they're also more sensitive to that, or when it's rainy and cold. So in general, collared flycatchers are a little bit more aggressive. They're dominant in fights, but they're also more sensitive to harsh environments. And because of that, they are strongly selected to breed earlier when there is climate change occurring. So this figure shows the number of breeding pairs in relation to the standardized laying date. So it's standardized across years. And the other curve shows relative fledging success. So you see that the peak in sort of fitness is earlier than the peak in the actual breeding date of the birds. So they are mismatched. They would benefit from breeding even earlier than they do. So even if they have adjusted themselves to a changed climate, they haven't done so enough. The picture is very different for pied flycatchers. So they are not as strongly selected for early breeding dates. So this would also show standardized laying date and the number of breeding pairs in relation to the relative fledging success. So this curve is rather flat, or with a little imagination, you might even see two peaks, but I won't go into details about that. So in general, a big difference between these two very similar species. And the very interesting thing is that we can see temporal isolation building up over such a short period as only 20 years. And what happens is that pied flycatchers males that are paired with collared flycatcher females, they tend to breed earlier than pied flycatcher males paired with the same species of females as themselves. So they avoid hybridizing by breeding later. So we see this connection between divergence in phenology and the buildup of temporal isolation between the two species. So to summarize this first section of my talk, divergence phenology can lead to temporal isolation. And this has been mostly studied in plants because it's kind of obvious that they can experience different microhabitats, even if they co-occur. But for animals that move around, it's more easy to think of temporal isolation as something happening in allopatry when they are experiencing different climates. But when they are together, it's kind of less sort of obvious to envision that there would be divergence in phenology. But we see this in pied and collared flycatchers. And we see that the species with a more narrow niche, the collared flycatcher, are more sensitive to trophic mismatch and therefore respond stronger to climate change. And then we have divergence even happening in sympatry and the buildup of temporal isolation. Okay, now we will change gears a bit, and I will talk about climate-driven divergence in physiology. Still the same two species in focus, and the possible link to the buildup of hybrid dysfunction. So this is the most popular model for how genetic incompatibilities can build up. And the idea is that this is something that happens when populations are experiencing periods in allopatry. So we have one ancestral genotype in a population. We have A and B, two loci that interact. And then we have a split. So the two populations are experiencing different habitats. And there is one mutation popping up in this population, big A change into little a. And this new mutation goes to fixation. In the other population, big B mutates into little b and goes to fixation, either because of drift or because it's selected or favored by selection in those environments. But when the two populations then come into secondary contact, little a and little b have never experienced selection together, so they work poorly when they are faced with collaborating, when they find themselves together in the hybrids. And in this way, genetic incompatibilities can evolve. Empirical evidence for that is largely limited to model organisms, because normally you would need model organisms to be able to study genetics in that great detail, which is needed in order to zoom in on genotypes that are on specific genes that can't collaborate. But we need more studies on this also in nature if we want to understand the gradual buildup of incompatibilities, but also the forces behind divergence in these genes to start with. And mitonuclear incompatibilities have been suggested to play a major role if we focus in on divergence in climate adaptation. And the idea behind this is that in order to have energy metabolism functioning, you need to have collaboration between two different genetic systems. So mitochondria, which is sort of the nuclear power of the cell, rely on their own genome and on genes in the nucleus, part of the genome as well, in order to have a functional energy metabolism. And that in turn influences the fitness of individuals across different environments, causing different selection, and then there would be changes in both nucleus DNA and in the mitochondrial DNA. And because these two systems need to collaborate, there is strong coevolution. And when we have hybridization going on, this sort of co-evolved gene complexes would be broken up. So back to the flycatchers. We have argued before that the reason why they differ slightly in life histories actually depend on past divergence in climate adaptation. And this is based on, if you look at the overall distribution of the two species, pipe flycatchers occur much more northern and have a broader distribution range. And collared flycatchers, the more sensitive species, has a more southern distribution and is more sort of narrow. They sort of more narrow in the niche. And this sets the stage for interactions between the two species when they co-occur. And as you've seen, also possibly future responses to ongoing climate change. And is this difference due to parents or their offspring? So why are nestling collared flycatchers more sensitive Is it because them themselves are more sensitive, or because their parents are less able to deal with harsh environments? And in order to figure that out, we did some experiments in the past when we actually swapped nestlings between the two different species. So we took pied flycatcher nestlings and put them into the nest of collared flycatchers and vice versa. And in that way, we were able to disentangle whether it was the parents or the offspring who was sort of to blame. And this was possible to do because they eat very much the same food. They are ecologically very similar. And we found that actually both pied parents and pied nestlings are better able to deal with harsh conditions. So my previous PhD student, Erin McFarlane, wanted to figure out more in detail why are collared nestlings more sensitive. And what she did was that she measured metabolic rate using this machine. So you can see a little nestling sitting here and there. And here is an adult male also in the chamber. So what we measure is actually oxygen consumption. Okay, so jumping directly into the results. What she found was that only pied flycatcher nestlings did adjust their resting metabolic rate to the temperature they experienced. So when the mean temperature was high, they had a lower resting metabolic rate as compared to when the mean temperature was low, then they had a higher metabolic rate. And that would be sort of expected based on what would be adaptive to do. So if you don't want to freeze to death, you should be able to have a higher metabolic rate when it's cold. But collared flycatchers didn't change their resting metabolic rate in the same way, so they remained unplastic. Similarly, when we did some experiments, so we did experiments where we moved nestlings between nests to sort of either reduce or increase sibling competition. And this figure would show enlarged broods where we have added nestlings to the nest, or reduced broods, or controlled broods. And the idea is when you enlarge a brood, put one or two extra nestlings in there, there would be higher competition for food, so less food per nestling. And also in this case, we found that pied flycatcher nestlings were able to adjust themselves to these new situations. So if they were found in an enlarged brood, they would have lower resting metabolic rate as compared to when they were found in reduced broods. And the idea is that this will also be adaptive, because when you are in a better condition, like more food around, you should have a higher metabolic rate, so you more fastly can put this sort of transform that into fitness, like you grow, you use the food. But when there is a poor food situation, it's better to have a lower metabolic rate so you don't starve to death that quickly. We also found some evidence that resting metabolic rate would be a little bit disturbed in hybrids. So this would be adult males that had been measured. And what we found was that the hybrids have much higher resting metabolic rate than the purebred species. And this sort of deviating metabolic rate will be costly if everything else is sort of constant. So when hybrids deviate from the parents like this, it's normally interpreted as something bad is going on. So digging a little bit deeper into this. So what would be the genomic basis to divergent climate adaptation, and possibly also hybrid dysfunction in this case? As I mentioned before, the mitochondria would be very important. And the OXPHOS pathway would be how they produce energy. I will show you that soon in a figure. And this part of the work was done by Erin McFarlane again, and also a very clever master student named Eva van der Heijden. And so we wanted to zoom in on the genes involved in the phosphorylation pathway, which is, so this figure shows this sort of pathway schematically. So you have the inner membrane of the mitochondria and the pathways, how ATP is produced, how energy is produced at the cellular level. And this is a process that is very well known actually on the molecular level. And it's also well known what genes, what mitochondrial genes and what nucleus genes are involved. So that is very nice to have sort of candidates to start with. So what we did was just wanted to see if there has been some divergence between the two flycatcher species in these genes that are relevant for energy production. And here you see the different complexes in the OXPHOS pathway and the sort of genes. And it's very few genes that are coded for in the mitochondria, which makes it also a little bit easier. So we sequence a little bit over 200 flycatchers, and then we zoomed in on all these different genes. And what we found was that there were a lot of non-synonymous mutations that had been fixed between collared and paired flycatchers. So there are some real clear haplotype differences between the two species. And here you see some preliminary pictures. So this is work that hasn't been published yet. So this is some preliminary work that I show you now. So here would be the different haplotypes I was talking about. So we have pied flycatchers down here and clusters of collared flycatchers here. And it's actually two different haplotype clusters within collared flycatchers. And this would be the correspondent haplotype networks for nucleus OXPHOS related genes. So also here there is some clear divergence between the two different species. So the take-home message from this second part of the talk would be that we see differences in adaptive plasticity of nestling resting metabolic rate between collared and pied flycatchers, and collared flycatchers are less plastic. So their nestlings are less able to respond to different environmental conditions, both natural occurring environmental differences, and also when we have experimentally manipulated the environment they are experiencing. And hybrids appear to have distorted resting metabolic rate. There is also some clear genetic divergence, both in mtDNA and in interacting nucleus genes. So a lot of pieces of information are sort of falling nicely together in the flycatcher system. So is there anything left for us to do? Right. A major next goal would be to actually reveal the link between divergent ecological adaptation, in this case, differences in climate adaptation in the two flycatcher species that have occurred when they were experiencing allopatric conditions, and if there is a direct link to the buildup of genetic incompatibilities. We saw some disturbed resting metabolic rate in the hybrids, which could indicate that there are some possible link here. And in order to reveal this, we would need to go down to the molecular basis to actually look at divergent climate adaptation and see what exactly genes are involved. And the same for possible clashes in the hybrids. And in this case, the candidate would be mitonuclear clashes in the hybrids. So the next step would be to go into individual details to look at variation in specifically interacting mitonuclear genes, and then see how that is linked to function of mitochondria. And in birds, they have functional mitochondria and blood samples, so we could do this non-invasively in a large number of individuals. So that would be the next plan. And then we can link that to the fitness of the birds in the different environments they are experiencing. And do this in both species, and we can also do it for the hybrids. So then it was possible to actually test this particular link. But that is something that is plans for the future. So I hope to tell you more about that in future talks. And then I would like to end by thanking my research group and all fantastic field teams and collaborators. And this is a picture of the field team for one season and some of their friends. So my group isn't nearly as big as this. So we actually are a quite small group like right now. So if you're looking for a postdoc, you're most welcome to join us. So thank you for listening.
Thank you very much, Anna. That was a really interesting and thought-provoking talk. Very nice and clearly presented as well. So while we wait for some questions to come in from the audience, I had a couple of questions myself that I'd like to ask you. So the first question is related to the first half of your talk when you were looking at the divergence in phenology and the temporal changes. I was quite curious whether because it was the collared flycatchers that responded more strongly to the change, and so whereas the pied flycatchers didn't change so much. And I was curious whether you have any evidence that that was directly linked to change in the prey dynamics at all, so the caterpillars that they were feeding on, and whether it was linked to that mismatch you were talking about, and so they were switching to match the switch in the caterpillar dynamics.
Mm-hmm. So your question is whether we have directly measured caterpillars. So we haven't done that particularly for all the years, but we did measure it for 2 years. And we have also related sort of the peak in caterpillar abundance to different tree species. So the peak in caterpillar abundance is a little bit steeper, or well, actually very much steeper in the deciduous forest. So we have a much higher peak there, but it's also more narrow. So in the more mixed forest, it's a lower peak and it's more stable across the season. So you see this big sort of the mismatch is much higher in the sort of rich deciduous forest than in comparison to the more mixed or poorer forest. But we didn't go out and measure the caterpillars every year. But what you can do is actually to measure also the phenology of the trees. So once you know how the caterpillars respond exactly, you can also go back and look at the green up maps of trees and do that in more detail. So that is something we're actually planning for the future. So thanks for that question.
Oh, wow. That sounds fascinating. And I was curious whether, if there's less of an overlap in the temporal occurrence of the two species, whether you think that could influence the proportions of those species in the population, because I was wondering whether if they're competing less in the same temporal window, whether that might influence the population dynamics so that the pied flycatchers increase in their proportion in the population, perhaps?
That's a very good question. I think if they time it right, if they make it to the really big peak, then there isn't very much competition over the food because they have well enough for everyone. But if they time it a little bit badly, then there would be stronger competition for what food to use. And it's also different between the two different habitats. So the divergence in timing of breeding is actually mostly seen in the more sort of mixed forest, where collared flycatchers still breed very early, but pied flycatchers are a little bit more relaxed, which is actually the best thing to do when you don't have this early peak, which complicates the story a little bit, because then pied flycatchers actually are better able at tracking the peak in the poor environment.
Ah, I see. So it's more complex, the interaction between the environment as the temporal. Yeah. That's fascinating. So we have a question coming from our audience, which asks, so as far as I remember, there's some possibility of back crossing in hybrids, as in F1 are not totally sterile. If this is the case, do you think there's potential for any adaptive integration of metabolism related genes between the two species?
Yeah, so in the past, there was some sort of evidence for backcrossing occurring, but we are not really sure whether they actually can backcross today. So there have been some gene flow between the species in the recent past, but I'm not sure if that is sort of totally cut off now, or if it actually can be some backcrossing occurring still. So we haven't been able to detect backcrossing yet on Öland, but I'm still hoping for that, because it would be very exciting, of course. But the question was whether there could be some adaptive backcrossing, and that is actually possible. So even if we have very strong selection against F1 hybrids, there could be some genes slipping by into the other species that could sort of be like you gain adaptations by stealing that from someone else. So in that way, it would be good for collared flycatchers if they could steal this tolerance from the pied flycatchers who have occurred for much longer in these more northern populations. So for collared flycatchers, there are at their northern limit to the distribution. So of course it would be a shortcut if they could steal those genes from the pied.
Yeah, that would be really interesting. It would almost be a fast track way to gain in some of the beneficial characteristics. That's great. So we have another question related to the phenology side of things. And the question asks, with migration, do you have an idea what triggers the migratory timing in the flycatchers, and could it be environmental or genetic?
Oh, good questions, but very tricky. So it would be difficult for them to know what the weather situation or the climate situation would be up here when they start from Africa. But potentially they could use some cues that are sort of more global cues. So I wouldn't like to exclude that possibility. But I think it's mostly genetically determined. That would be my guess. But that would be only guessing and not something I know.
That's interesting. And the question actually speculates, so if there was also some environmental influence, could the overwintering area have a bigger influence over when the birds leave, for example, because there's evidence that hybrids have an intermediate timing, and would that contribute to higher success because they could potentially make it into each species breeding peak?
Yeah, so I guess that's a good idea from some other species, but the pied and the collards are so genetically diverged. So the hybrids have such a poor quality. They basically almost totally sterile. We have very little evidence for still ongoing gene flow, so they're very strongly selected against. So I think it would be more likely in some other cases than in this case.
That's interesting. Thank you. Okay, so while we see whether there's any more questions that are coming in from the audience, as I think people need a bit of time to just process, so for the questions. I was curious whether you see any sex differences in the offspring in terms of their ability to withstand the harsh conditions, or whether that sex doesn't seem to play any effect in these patterns?
So I haven't looked into that myself, but there has been some studies in the past looking at sex differences in how they tolerate change brood sizes, and they didn't find any evidence that one of the two sexes would be more influenced than the other one. I mean, some expectations would be that males are more sensitive, for example, but that didn't happen.
That's great. Thank you. That's interesting. I'm really curious about your future work as well, and exploring more of the dynamics from the gene sequencing data. I think that's quite exciting to look at the interactions, the effects on fitness as well. And I don't know whether you have any early indications of what might come out in terms of the effects on mitochondrial function from the sequencing data that you generated.
I wouldn't like to guess anything. I mean, the indications from the phenotypic work we've done so far would be that it has a big influence, but I don't dare to say anything before I've checked it.
That's great. Watch this space for the next installment. That's great.
Yeah, and we're also planning to zoom in. So in some cases we have, fixed differences between the two different species, but you would have a fixed no variation in one species, but variation in the other species. And then it would be very neat to zoom in what the variation does in the species that is not fixed for that.
I see. More investigation on a more species-specific level, I guess.
Yeah.
That's great. Thank you. So I'll just see if we have any other questions coming in at the moment. So I think that's all we have time for today. Yeah. But yes, thank you very much, Anna, for a very fascinating and interesting talk. Thank you.
Yeah, thank you for inviting me.
That's great. So I'd just like to remind everyone that our next talk will take place next Wednesday and will be presented by Professor Scott Edwards from Harvard University. So we hope that you can join us then, same time and same place. And in the meantime, check out our updates on Slack and our Twitter feed and spread the word about our seminars. Thanks for taking part, and until next time, stay safe and see you again soon. Goodbye.